Next, cytokinins. Cytokinins have a specific effect on cytokinesis, that is the division of cytoplasm in the formation of the two cells, that is in cell division. And it was discovered as kinetin from herring sperm DNA, autoclaved herring sperm DNA. The kinetin do not exist naturally in case of the plant. So discovery of natural substances having growth properties led to the discovery of the gelatin from corn kernel as well as coconut milk. After that, several synthetic cytokinins are discovered, all the nothing but having components of adenine derivatives like benzoyl aminopurine, benzoyl adenine, which are synthetic cytokinin discovered having adenine components, derivatives of adenine components. Next, natural cytokinins synthesized more in rapid cell division areas like root apices, shoot buds, young fruits which are green in color. And cytokinins are responsible for new leaves, chloroplastin leaves, lateral shoots, and adventitious shoots. Adventitious roots, auxin. Adventitious shoots, cytokinin. It delays the senescence, it postpones the senescence, mobilization of the nutrient. That means it prevents chlorophyll degradation. Senescence is yellowing of the leaf, ultimately leads to death of that organ, the leaf. What do you call it? Is senescence. So it delays the senescence, never prevent the senescence. It delay or postpone the senescence by mobilizing the nutrients. Next, cytokinin is antidote for decapitation. That is, it removes apical dominance. Without cutting of the apical bud, if you employ the cytokinin, apical dominance is going to be removed. And the cytokinin having the cell division dividing properties, that is along with auxin used in tissue culture. So cytokinin and auxin have a synergistic role in case of tissue culture by inducing cell division. Cytokinin and auxin having antithetical properties with regard to apical dominance, as apical dominance is favored by auxin, removed by cytokinin. Next, ethylene. Ethylene is simplest gaseous phytohormone produced by almost all parts of the plant body. Especially it is synthesized in senescing organs and ripening fruits. Ethylene causes triple response growth, that is in seedlings, horizontal growth of seedling, swelling of axis, that is lateral thickening, and apical hook formation. And ethylene responsible for senescence and abscission of leaves and flowers. And ethylene much effective especially in case of fruit ripening. And it enhances the rate of respiration during ripening of fruit. Such a rise in rate of respiration during ripening of fruit, what we call respiratory climactic. Next, ethylene breaks the dormancy and promote seed germination in case of peanut and promote the sprouting of potato tubers. Next, ethylene helps the plant to increase the surface area for absorption of water because it is responsible for root growth as well as root hair formation. Next, in deep water rice seedling, upper part of the shoot above water surface, this is ensured by ethylene induced petiole and internodal elongation. And ethylene is one of the PGR widely used in case of agriculture and horticulture because it is having wide range of physiological applications. And ethylene is responsible for synchronous flowering and fruit ripening in case of the pineapple. So number of pickings reduced, even acres of the land, the fruit harvesting is simultaneous. Next, the most widely used ethylene is ethifone, that is 2-chloroethyl phosphonic acid. in aqueous solution, readily absorbed and distributed to all parts of the plant body, releases slowly. It is responsible for ripening of fruits in case of tomato as well as apple. The sithiphon responsible for abscission of flowers and fruits and thinning of fruits or pruning of fruits in case of cherry, walnut as well as cotton. Because as it involved in abscission of the flowers and fruit, to the rest of the fruit more nutrients are available, large sized fruits are present. So it is a commercial application. And it increases the yield in case of the cucurbit because it induces pistillate flower formation and also responsible for flowering in case of cucurbits. And next it is abscisic acid. Abscisic acid has a role and discovery was attributable to abscission and dormancy. Like other PGR, it is also having a wide range of applications in the plant growth and development. Especially it's the inhibitor of the growth, inhibits seed germination, inhibitor of metabolism. And the ABA synthesized in the leaves leads to closure of stomata. So increase the tolerance to abiotic stresses. That's why it is considered to be a stress hormone. And it is responsible for seed development, maturation, and dormancy. Because it by imposing the dormancy, it prevents the seed to avoid the conditions like desiccation and unfavorable conditions for growth. In most of the situations, ABA act as antagonistic to gibberellic acid because gibberellic acid promote germination. ABA induce, that is inhibit seed germination. So in phases of the growth, whether development, the growth or differentiation, the one or other PGR may interact to affect the physiological process. So we may not attribute this phytohormone is concerned with this function because whose actions may be individualistic, complementary, synergistic, antagonistic. Suppose if we take ethylene, fruit ripening, it is individualistic. Auxin and cytokinin together promote cell division, it is synergistic. With regard to seed germination, promoted by gibberellic acid, inhibited by ABA, antagonistic. And if you take auxin initiate root, cytokinin initiate the shoot, whose effect it is to complete the process. One start this, other completes this. That is complementary. So at any stage, the one or other PGR may interact to produce the effect. And at many times, diverse PGR produce the single effect and similar one physiological effect caused by diverse PGR. Suppose the senescence, they caused by ABA as well as ethylene. Dormancy, ethylene as well as it is ABA. And apical dominance promoted by auxin, removed by it is cytokinin. So we cannot attribute the one function to one hormone. Next, the PGR is one of the intrinsic control only, not only the intrinsic control. Because in intrinsic control, internal control, intracellular, intercellular. Intracellular is genomic, genetic control. Intercellular is the chemical. So other factors like it is external factors. The external factors like temperature and light affect the development, differentiation, the growth via PGR, plant growth regulators only. Like vernalization, low temperature treatment, a hormone vanillin is responsible. Photoperiodism, for flowering, the florigen hormone it is responsible. Like this, the external factors may also influence the development of the plant growth via PGR. I am signing off from this episode with the completion of the plant growth and development. And in subsequent episodes, the so many other concepts with other lessons with regard to biotechnology and molecular biology. Don't forget to subscribe and share to the others, those persons who are seriously preparing for NEET.